J'ai le plaisir d'accueillir aujourd'hui le professeur Sylvain Moineau, qui est microbiologiste et qui nous vient de la belle province, puisqu'il est professeur de microbiologie à l'université Laval à Québec. Et le professeur Moineau travaille sur les bactériophages. Et aujourd'hui, il va vous présenter un travail sur les CRISPR. Et donc, merci beaucoup pour présenter ce travail.
Merci beaucoup. Alors bonjour, merci de vous être déplacé. Merci de l'invitation également. Vous savez, j'ai quitté juste avant une tempête de neige de 30 cm. Alors vraiment, j'apprécie mon séjour à Marseille. Donc, vous allez devoir vous passer de mon accent québécois, puisque je vais faire ma présentation en anglais. Et bien sûr, si vous avez des questions en français par la suite, ça me fera plaisir d'y répondre. Ça vous va? So, this is the outline of my talk. I'll talk to you today about the technology, CRISPR-Cas9. Just show of hands, who is using CRISPR-Cas9 today? All right, we'll go quick. That one. And who has heard of CRISPR-Cas9? Otherwise, I would have gone right away. All right, so I'm going to talk to you about the headlines because CRISPR-Cas9 is probably something that people know about. And then after that, I'll go jump into the history of how CRISPR-Cas was discovered. And then I'll finish up how it works and what we're doing today using the technology. So this is the first slide that I like to start my presentation with. This is a number of publications that are related to CRISPR in the past 10 years. So the acronym CRISPR started in 2002, and this is 2018, and you can see this is going way up. This is what you would call a career opportunity. All right? So if you do not know CRISPR or you're not using it, maybe you should think about it. All right? Because this is becoming as popular as PCR. All right? So it really started, though, in 2012, where there's a big paper that was published by Emmanuel Charpentier and Jennifer Doudna's lab, where they showed that you could use CRISPR-Cas in a tube to modify a genome. All right? A few months later, and the months are important here. In August 2012, this is a big paper, and I'll show you why it's a big paper later. A few months later, another group in Boston showed that you could use this in cells. You could use the technology to modify the genome of a human cell line. All right? And then the field exploded after that. Pretty much everything that we know that is living organism today, including viruses, have been modified using CRISPR-Cas9. And look at the year those publications were published. 13, and human cells, obviously livestock, mice, flies, and so forth. Very rapidly, people start using the technology because it is simple. And it got adopted by many, many labs around the world. So essentially what you need, if you want to use the CRISPR-Cas9 technology, you need to modify a gene of interest. All right? And then you go look at the sequence of that gene. You go try to find this little motif, and I will explain to you what it is and how it was discovered. Then you need to design a GPS or a guide RNA that will go and target the region you want. Then you need the molecular scissors, which is a nuclease called Cas9, from Streptococcus pyogenes. And then you will let the cell repair itself. Because when the human cells is cut, at some point you need to repair the genome, otherwise it's going to die. All right? There's a trick there, where you can tell the genome, I want to repair, but you can repair the way I want. Right? And then you use the cell system to repair it, and if you provide a donor DNA, a template, you can actually introduce a gene. All right? So this is a video from MIT that we have edited with their permission, just to show you briefly the technology. So this is the nucleus of a human cell. This is obviously a cartoon. This is the DNA. I hope you recognize. This is Cas9, the protein, which is armed with an RNA molecule. It will scan the DNA and recognize this little motif called PAM. Then this was destabilize the double helix DNA. And then one RNA will actually, which is armed with the Cas9, will cut. The Cas9 will cut the DNA. And at that point, the cell needs to repair itself, right? And then if you provide a template, you will introduce biomologous recombination, for example, or homology-directed repair, you will introduce a gene. Voila. That's how it works. It's very simple, and that's why so many people are using it. All right? Of course, there are issues, and I will briefly talk about them later on. What people don't know as much, though, is that part of the technology, the parts of the technology, came from the study between phage and bacteria. All right? These key components came from those studies, and I will go back in history just to let you know how this was discovered. But before doing that, I need to talk to you about phage a little bit. This is what I call phage 101. I'm sure many or most of you know that phage or viruses are the most abundant biological entities on the planet. There's nothing more abundant on the planet than viruses. This is not human, no insects, or no animals. Viruses are the most important. You go to the sea here, take water in the palm of your hands, you got more viruses in your hands than there are humans on the planet. All right? So think about that the next time you go to the beach. So we're full of viruses too, by the way. All right? Every time you find bacteria, you'll find phage, you'll find viruses, and if you do not find them, it's because your protocol is wrong. Okay? Because you should find them. Today I will talk to you only about phages that are virulent, meaning they infect the cells and they kill the cell. And I'm only going to talk to you about double-stranded DNA viruses, or DNA phage. I'm not going to talk to you about RNA viruses, only double-stranded DNA. They come in different shape or forms. There are big ones, smaller ones. Some of them have tails, some don't. Their genome can be double-stranded, single-stranded, can be RNA, can be DNA. At Université Laval in Quebec, we have a very large phage collection. And that collection started in 1982, so it's kind of a biobank, much smaller than what you have here, obviously. But Professor Ackerman started this collection in 1982. He retired in 2002, and since 2002, I'm the curator of this collection. I've been at Laval since '96, but I took over the collection in 2003. And over the last 6 years, we have shipped phages all over the world, in 35 countries, in different 300 labs. The phage research is pretty much the same curve as the CRISPR. It's going up right now. There's a lot of people are interested in phages because we're realizing they're found everywhere. We can use them maybe in phage therapy purposes or biocontrol agents. Bottom line, phage research is going up. If you want to have controls in your experiment, phage, for example, you can call us because we'll probably have control phage for you. All right. We've got phage infecting 120-something bacterial species. And Denise Tremblay is the person in charge of the phage collection. So phage, they'll bind to the cell surface like any viruses. You need a receptor. This is something I call a lock and key system. So you need the right key at the virus end of the tail. You need the right lock here. And if there's a perfect match, the phage absorb. DNA is injected inside the cells. And this process, it's pretty fast. The virus I'm going to talk to you today do this cycle in half an hour. So every half an hour, this entire cycle is done. And the new virions, about 100 new virions, will be released into the environment, ready to infect other cells if the receptors are present. All right? These are like the Usain Bolt of virology, right? They're really fast. When they get inside, they take over the cell, and that's the end. Right? The virus will win. The virus will always win. Just to give you an example how fast this can go. If you start with one bacteria, half an hour, you've got 2 bacteria. This is basic microbiology. You should all know that. When you take the phage, half an hour, we start with 1, get 100. An hour and a half later, if optimized condition, you can start from 1 over 1,000,000. This can go really fast. So think about that if you do a fermentation, for example, or if you do a bioprocess with bacteria. If there's a phage in your environment, it can kill your bacteria pretty fast, and then you'll be in trouble, right? And the dairy industry is a big example of that. You know, if you eat your cheese, eat your yogurt, you look at the list of ingredients. Normally you should see milk, well, we all hope, and then you'll see bacterial cultures. So we're adding bacteria to milk, to transform milk, to make cheese, to eat cheese. Phage will survive the pasteurization process, so they will be present. If you add bacteria that are sensitive to these phages, you're in trouble. That's one of the reasons why the quality of your cheese sometimes is varying, because phage will kill some of these bacteria. So there's a very big interest to have bacteria that are resistant to these phages, okay? Luckily, bacteria have a bunch of defense mechanisms or anti-phage mechanisms. There are mechanisms that will block the phage to absorb. There are mechanisms that will prevent the DNA to get inside. Recent paper in Nature show that some bacteria are producing chemicals that will block the replication of viral DNA. There are systems that will cut. If you're using restriction enzymes in your lab for cloning experiment, for example, these were discovered because they were anti-phage systems. CRISPR-Cas is one of them now as well, and so forth. There's many, many different mechanisms the bacteria are using. Not one bacteria will have all of them, but globally, there are a bunch of defense mechanisms. All right? Otherwise, it'd be no more bacteria, and probably no more viruses, and probably not us as well. All right? So that's Phage 101. I hope you get a quiz at the end, by the way. It's a joke. So CRISPR, it stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR, so all together, cluster. And so cluster is the acronym that was actually discovered in 2000, or proposed in 2002. There was many more acronyms that were proposed, but for some reason, this one stuck, and people keep using it now. And I'm sure they're really happy about it. And Cas for CRISPR-associated genes or associated proteins. Okay? So what is a CRISPR? So this is a DNA sequence. This is a CRISPR. You have repeat sequences. So the same sequence that is repeated which are separated by variable sequences. Those variable sequences are called spacers. Okay? And this was discovered for the first time in E. coli. In 1987, a group in Japan was cloning a gene in E. coli, studying that gene, and they sequenced it. Back in 1987, you get a PhD by cloning a gene and sequencing it. That was it. Now you can do this in an afternoon, right? But back in the days, this was a very big deal. Turned out when they were sequencing it, they actually noticed that next to it, there was this unusual nucleotide arrangement. So they saw that in the genome of E. coli, but they did not know the function of it. So it stuck there for a while. People didn't know what was the function, but they were using a little bit because those variable regions here were actually strain specific. So one E. coli will have this set of spacers that is specific to that strain. So a lot of people were actually using the CRISPR to track strains. All right, so they were systems that were devised using the spacer sequences. Repeats are really short, between 20 and 50 base pair, depending on the bacterial species or genus. In Streptococcus, which is the bacteria I'll talk to you about, is about 36 base pair. Again, species specific. It will vary between two repeats, which are here are black diamonds. You got a repeat, a spacer sequence. The rectangles indicates different sequences, and you've got the diamonds where represent the repeats. There's even one strain out there in the literature that has over 500 repeats. If you can believe that, in the genome of bacteria, over 500, all with different spacers in the middle. All right, so this can be stable also in the genome. Spacers also are fairly short, between 17 and 85, depending again on the bacterial species. In Streptococcus, it's 30 base pair. So a repeat spacer unit is about 66 base pair in Streptococcus. Okay? Again, strain specific. In 2005, 3 papers came out, 2 from France actually, showed that these spacers, those variable sequences, some of them, not many, but some of them were matching viral sequences, were matching plasmids, were matching mobile elements. So the first hypotheses came up. Well, maybe CRISPR is involved in some sort of defense or some sort of interaction between virus and bacteria. But these were hypotheses, right? So that's a CRISPR. Repeat spacers matching viral sequences. Next to it, you've got the CRISPR array here. Either upstream or downstream, you'll add genes. They're called CRISPR-associated genes. And this is what makes this field super interesting. Because depending on the bacterial species, the genes that are associated with the CRISPR array will be different. So that's why now we have 6 big types, numerous subtypes, and they will work differently because it's not the same proteins that are involved in the mechanism. All right? So that's the only extent I will talk to you about. There's a lot of variation, and the field got Let's say in the 2010, 2011, in the literature it was a big mess because people did not know how to call these things. And Eugene Koonin and a bunch of us, we came up with a classification of the Cas genes to make sure that people will use the same nomenclature, because people were starting to find a CRISPR, oh, I'm going to call this whatever name. And it was very hard to have some sort of consensus on the naming of these proteins, right? So there's this nomenclature now. But today, I'm going to talk to you only about one type, which is a type 2. The smallest one, the most simplest one, and the one that has Cas9, which is used for the genome editing. All right, so Cas9, Cas1, Cas2, which are universal, and almost, we used to say universal, but now it's almost universal, and Cas4. And of course, all these proteins have been crystallized. We have a pretty good idea of what they're doing, like especially Cas9, of course, which has been used quite a lot, modified a lot. I'm going to move on this. If you're interested, you want to know if your bacteria has a CRISPR system, you can obviously sequence your bacteria, but you can look on this website in Paris Sud, and they have annotated abundant genome, and you can look and see if they are already CRISPR present. The database will tell you right away. For example, here Strep pyogenes has one CRISPR, and you can click on it and it will tell you what it is. So it's really neat. Sometimes you'll find questionable structures. When it finds questionable structure, it means there's no CRISPR. All right. And of course, it might be none whatsoever. So if you're interested by the field, that's a good start. You can go to this website. All right. So, but how does it work? What's the function inside of bacteria? Now we know what it is, but back in the days we did not know, right? In 2005, 2006. And now we know it works in 3 main steps. So again, in the bacteria. We're not talking about genome editing here. We're right inside the bacteria, all right? How it works. It works in 3 steps. First steps, we call it adaptation, or immunization, or vaccination, and then we'll show you why. Second step, expression. We're going to generate RNA molecules. Cas proteins. Third step, interference, where the Cas proteins and the RNA will bind together and then they will target DNA and cut it. All right, so 3 steps: adaptation, expression, interference. The genome editing part, use only that part. All right, but in the bacteria, obviously you need those 3 steps. And to demonstrate how this was discovered, the function, I'm going to use a bacteria that you might have eaten this morning. If you ate yogurt, you probably ate that bacteria today. All right? So Streptococcus Thermophilus, this is the name of the strain. This is a very popular strain used around the world to produce yogurt. Why it is popular? Because it's producing a very nice polysaccharide, which gives you a very nice mouthfeel. All right? You do not need, for example, to add ingredient like pectin or whatever, or gelatin to make your yogurt like more thick. So it's producing natural polysaccharides, right? Very important strain. However, sensitive to phage. So the idea here was to make the bacteria resistant to phage without GMO, because you want to eat that stuff in Europe. US, they don't care. But here, kind of important. All right? So we want to use these things that are natural. Turn out, we have the genome of that bacteria, and it carries CRISPR-Cas system. So it was a perfect model to test what would be the function of these systems. My lab has been studying phage for a while. We sequenced our first phage genome in 1997. I don't know, some of you might not have been born in 1997. But anyhow, in 1997, sequencing a phage genome in Canada was $40,000. So maybe, I don't know, 20,000 euros, something like that. Now you can do this under 100. It's insane. So in 97, we started to sequence phage genomes because we thought this would be very important. So we had a bunch of phage infecting Thermophilus, including that strain, that we had a genome. Okay, so these were our bases. And where it works, and I'll show you how we do this, is the phage will get inside, the genome will get inside, Cas1 and Cas2 will form a complex, and it will pick up a piece of the phage genome, introduce it into the array at the 5' end of the array, and this repeat will actually be duplicated. All right, this is how it happens. And how we show that. So this is a plate that I'm sure you've all seen. This is a lawn of bacteria. So if you add a little bit of phage, you get plaques. You've seen that before, right? No? Please, yes. If you have many phage, so of course the phage have infected the cells, new variants have been released into the environment, they have infected surrounding cells, so it's a chain reaction, right? And what you see is the end of the lytic cycle of multiple replication is one plaque. So if you had multiple phage on your plate, the plate will be almost, the bacterial lawn will be almost gone because the phage will take over, right? It will kill all the cells that are on the plate, right? And if you had a lot of phage, there's no more bacteria. But if you over-incubate these plates, 2 days, 3 days, you'll start colonies that will start to emerge. And those colonies are phage-resistant colonies. They're natural phage-resistant colonies. If you do this with E. coli, most of the time it's the receptor at the cell surface that is mutated, right? I was trying to show you at the beginning. But in Streptococcus Thermophilus, the receptor is the same. So the phage DNA was getting inside the cell. Something was happening inside the cell. Again, we had the genome of the bacteria. We designed primers that were targeting the CRISPR array. So we pick up colonies, run PCR. We had primers here to amplify the various CRISPR arrays in the strain. And sure enough, every single colonies we picked, when we PCR the CRISPR array, we had a change in size. It was insane. I remember a postdoc coming in my lab, and you're not going to believe this. So we had an increase of PCR product, 66 base pair. You see where I'm going, right? So that 60 extra base pair, there was 36 was the repeat, and 30 base pairs was different. And sure enough, it was coming from the phage genome, the phage that we use in a challenge assay. So essentially, you pick 2 colonies here that are phage resistant. You compare to the wild type that is phage sensitive. It has a CRISPR array already, but it will have acquired at a 5' end a new piece of DNA from the phage genome, and this repeat again has been duplicated. And if you take 2 colonies, and if you take 10 of them, that piece of DNA will come from different region of the phage genome. I don't know what gets you excited about science, but this is pretty cool. You know, CRISPR can pick up piece of DNA from a phage genome and use it, and I'll show you how it works to defend itself. Beauty of it, we've got multiple phage. We can do this again with another phage that is different than the previous one. We do the same experiment, take one of these colonies that were resistant to phage, use another phage, and do the assay again. What do you think is going to happen? Another spacer will be acquired. It will keep adapting and adding new spacers like that. We have done this using at least 10 phage. We keep adding up new spacers. All right, so the strain becoming more and more and more phage resistant to a number of different phage. All right, so this is what we call an adaptive immunity because it's adapting to the phage it's facing. And now it became a sort of the spacers became, now that we know this, the spacers are now what we call a kind of a molecular archives. A molecular archive of past infections or past encounter of viral infection. Of course, if you have more spacer, you're more phage resistant. Skip that part. So then we were looking, why this piece of DNA is picked up? Why not something else? It was not all the piece. It was not all spot in the phage genome that was acquired. Some regions. Why those were acquired? So we're looking at the DNA that was acquired as a spacer. We tried to find a motif. Could not find anything. We're not too bad in bioinformatics, but we could not find anything. But if we go back on the phage genome and look at that piece of DNA that came from there, what we call now a proto-spacer, meaning comes from, if we were looking next to it, so this is a spacer that was acquired in gray region here by one colony. This is another spacer that was acquired by another colony. But if you look at the phage genome, the same sequence was present, but next to it, there was this little motif that was present all the time. And this became the proto-spacer adjacent motif. All right? And we'll see how it's important later on. So that motif, for that piece of DNA to be picked up by the CRISPR array, next to it, you need to add that motif there. And if you can believe this, in the phage genome that we use for the challenge assay, that motif here is found 233 times. Meaning that one phage, one bacteria, you could, in theory, get 233 different colonies that has picked up different pieces of DNA. That's what we call it vaccination or immunization. It's pretty amazing. Well, I like to think it's amazing. All right. Now we know that the PAM will vary depending on the CRISPR-Cas system. Strep pyogenes, it's a smaller PAM, and so forth. And this is what it is used for genome editing. We know it's an NG motif. So again, in the bacteria, you've got a repeat, spacer, repeat. In the phage, protospacer, the same sequence. And next to it, you've got the PAM, which is not present there. So this is a way for the bacteria to differentiate between itself and non-self. Okay, the PAM is important there. So spacer in bacteria, protospacer in the phage. But if you've been working with phage, like I've done for a long time, there's a problem here. I was telling you that those viruses are very fast. How in the world can a phage get inside the cell? Cas1, Cas2 has enough time to go pick up a piece of DNA, put this into this CRISPR array, and voila. It doesn't make any sense. Well, at least we don't think it does. So that's why we came up with this hypothesis where we think that this was happening only when the cells were infected by defective phages. You know, when I was telling you we've got a bunch of viruses in our hands, well, not all of them are actually virulent. Some of them are defective. That's the nature of things. You know, not all viruses will be functional. Some of them will be chimeras, will be just non-functional, natural non-functional. And Alex Hines, postdoc in my lab, ran this assay where he actually treated the phage population with UV, or restriction enzyme, just to cut it out. And then do the challenge assay again. And sure enough, by using defective phages, the number of colonies went way up. We're picking up way more colonies that have acquired new spacers. So we think that this is happening only when the cell is infected by defective viruses, which, by the way, is really reminiscent of when you're getting your flu shot, right? When you get your vaccination, we're getting flu shot from inactive viruses. The bacteria might have been doing this for a long time, all right? So when we write these papers, we always get fun sometimes. Anyhow, so it's the same process. It's defective. We think it's just defective. All the molecular details associated, or most of the molecular details that are associated with the integration of new spacer into the array have been resolved, thank you, resolved at the molecular level. And great work done by Jennifer Dowler's lab. Essentially, it works like an integrase, right? It's integrating new piece of DNA. I don't have the time to talk to you about this, but CRISPR array can pick up DNA from plasmids too. All right, not only phage, but from plasmid as well. And the strain can actually pick up from self-replicating plasmids. And when it picks up a piece of DNA in the CRISPR, that strain is at that time resistant to that plasmid. The plasmid is gone, and if you try to reintroduce that plasmid, you can't. Okay, so that's what we're calling them PIM, for plasmid interfering mutants. Those bacteria are resistant to the plasmid. So you just put the cell with the plasmid, grow them with antibiotics, remove the antibiotics, for example, if there's a marker in there, remove the antibiotics, and wait for the plasmid to get lost. Take those colonies that have lost the plasmid, check with a CRISPR array, and sometimes you'll see this, right? It was really cool, though, when Josiane Garneau in my lab did this experiment. She had an antibiotic-resistant gene into her plasmid. And it turned out that some of the colonies that acquired a piece of DNA from the antibiotic-resistant gene, and that strain at that time was resistant to the reacquisition of the antibiotic-resistant marker, which was pretty cool, and these they liked these things. So, bacteria with an active CRISPR-Cas system can acquire new spacers from foreign DNA such as phage genome, plasmids, mobile elements, and so forth. What I've told you so far, now for the past 5 years, our students, seniors in microbiology at Université Laval are redoing this all over again. So they're using phage, bacteria, generating colonies, PCR, sequencing, looking at where it came from, and so they're redoing the same experiment, so it's now becoming part of our undergrad study in microbiology. So students, they like these things, so it's pretty cool. We really like this experiment, and again, it's running out right now for our 6th year, and it's working great. So everything has been published, so if you want to use it in your own lab, we have shipped the phage and bacteria to many labs so they can try it, and it's apparently working in other countries, so it's always good to know. Second steps and third steps, I'll be quicker, don't worry, so you can go to sleep. Expression. All right, second step. C'est une diapo en français. So sorry about that. Intermédiaire en X, voilà. So CRISPR, you got the CRISPR array. There's a promoter here, so this will be transcribed. So you get a long RNA molecule with the repeat spacer, or complementary repeat spacer, repeat, so forth. This will be processed. And at the end of the day, you'll get CRISPR RNA, so small RNA molecules that will be matching part of the repeat and part of the spacer. So inside the cell, you get this long RNA molecule, but you get also these small RNA molecules, okay? And in type 2, because the system, the way the RNA will be processed will vary from types to types. So I don't want to get into different types. I'll focus only on type 2. But you have to know that if it's a type 1 or type 3, the RNA processing is different, okay? But in type 2, and this work was done by Emmanuelle Charpentier when she was in Sweden at the time. What Emmanuelle, so again, in streptococci, promoter there, you get RNA molecules that are between 38 and 42 nucleotide. But upstream here, she found, because she was running RNA-seq experiment, she found other RNAs. And she also realized that some of these RNA were actually matching part of the repeat. So the bacteria will actually generating two kinds of RNA molecules. The CRISPR RNA and something called trans-acting RNA. And because they were matching, it turned out that this molecule here, very small molecule, will actually provide a double RNA molecule with part of the repeats matching the tracer and the other part matching the spacer would be free floating around, ready to bind to something else, okay? And this would be processed also by the RNA3 of the host. But at the end of the day, we're generating this RNA molecule with the CRISPR and the tracer RNA. So what it means, that tracer RNA was actually the handle to bind to the Cas9 molecule, that we know. So inside the cell, you get the Cas9 here, which is a blob here in light blue. You've got the CRISPR RNA molecule matching the spacer, the repeat. You've got this part matching the repeat, and this part, which is used to bind for Cas9 to actually hold on to the CRISPR molecule, to the RNA molecule. And this is inside the cell. This is what you call the surveillance complex, all right? Ready, just like almost acting like an antibody. It's obviously not an antibody, but almost like. And why some people would get a Nobel Prize here is because they had a very neat idea. Instead of dealing with two RNA molecules, they decided to fuse them, just adding a little linker here. And by just using this all the time, but changing this, you can target any piece of DNA you want by base pair with RNA and DNA. You just need to change that. And then you can, and of course, you need to take into consideration the time, but you can go and bind a piece of DNA. Following me here? Or, no? That's all right. At the end, hopefully it will make sense. Third step, so again, spacer acquisition, RNA production, binding to Cas9, and then third step, cut the DNA. So how we did that was pretty simple. We had those colonies that had acquired spacers from the phage genome. We grow those cells. This is the wild type that is sensitive to two phage, as you can see here. We take another colony that is resistant to one phage, sensitive to the other, and then you've got this colony that is resistant to two phage because it has acquired spacers, right? So we took those cells, we infected them with phage, and then we look at what's happening inside the cell, the phage genome, okay? This is the sensitive, so this is the phage genome, about 30,000 base pair. We focus on that region of the phage genome because the protospacer is localized here. So we infected the phage sensitive cells with phage and follow over time with probes using this big old southern assays. And you can see over time, 2 minutes, 15 minutes, the DNA is replicating. The phage is replicating inside the cell. So it's a phage sensitive cell, so it's replicating. But in the one that has acquired new spacers, this piece of DNA was emerging. Turned out this was DNA of the phage that was cut. right? So we had enough DNA to sequence that. So these are different colonies that acquire different spacers. This is the proto-spacers that was found in the phage genome. And sure enough, it was always cut 3 nucleotides next to the PAM. Always cutting at the same place. 3 nucleotides next to the PAM sequence. So if you inactivate Cas9, there's no more cut, indicating that Cas9 was involved in the cleavage activity. So what you get is you've got Cas9 here, again in blue. You've got a tracer in the guide RNA inside the cells. There's no linker in this case. And then you get the target DNA by base pairing with the RNA molecule, or the RNA part that is matching the DNA. It will cut bluntly 3 nucleotides next to the PAM. Voila, this is how it works inside the bacteria. I'm going to skip this. So this is so Cas9 has been studied to death because people are interested in this. This was shown at a CRISPR meeting in 2017. So it was the first time that we actually saw live Cas9 cutting DNA. Group in Japan using atomic force microscopy. So this is Cas9. You can see a bit the blob. This is a very big protein, 1,200 amino acids, and this is DNA. And at some point, you see the DNA is being cut. That was a CRISPR meeting. It was in the US, and then you people in the crowd like, ooh. We're seeing for the first time in real action Cas9 cutting DNA. So it's really cutting DNA. All right. So again, inside the bacteria, 3 steps: adaptation, vaccination, acquisition of spacer, expression, production of RNA molecule, which will depend on the type. And at the end of the day, it cuts DNA. And that the phage cannot repair itself. When its DNA is cut, that's the end of it. All right, the bacteria wins. And you get yourself a bacteria that is resistant to phage. So this is a cartoon video that kind of shows what I was trying to tell you. So this is a yogurt bacteria having fun multiplying in milk. It's just a Streptococcus. Now, unfortunately, it's going to be infected by the phage. So you know the story here. The phage takes over the cell, starts producing its own proteins, self-assemble. This is nanotechnology at its best. And after 30 minutes, the infected cells will be blowing up because the phage wins, right? And those phage will infect other cells. However, in some cases, those cells will be infected by defective phage. DNA will get inside the cell. Cas1, Cas2 will recognize that, will scan the DNA, be able to tuck a piece of that DNA, introduce it into the CRISPR array, and at that time, that cell is now resistant to further infection by the same phage. So it's producing RNA molecules, producing Cas9, and if another phage infects the cell, it will be recognized by a surveillance complex, and Cas9 armed with the RNA molecule will cut the phage DNA. Game over from the phage, the bacteria wins, we eat yogurt and chew the There's always a but. Phage, again, the most abundant biological entities on the planet. So if you're the most abundant biological entities on the planet, you need a few tricks. Otherwise, you'll be lost. You probably not see that at the back because you're sleeping, but there was here this little plaque there. This is what we call a phage mutant. Right? And when you're studying phage or viruses, you love mutants because these are really great biological material. So we took those plaques there, sequenced the entire genome, 30,000 base pairs. We found 1 mutation. It did not cost $40,000 to do that one, so it was much cheaper. 1 mutation. What do you think it was? It was in the protospacer region or on the PAM. So there's 3 examples, 3 different mutants here. So this is the spacer acquired by the bacteria. This is a protospacer in the phage, the PAM region, and sure enough, it had mutations in the pro. So 1 single mutation is enough for a phage to bypass the system for 1 spacer. Right? That's not very efficient. Of course, if you add 2 spacers, that's different. You need the phage to have 2 mutations. A bit more difficult, more rare. And then came this paper from the University of Toronto in Canada, where they found in Pseudomonas phages proteins that were actually inactivating the CRISPR system in Pseudomonas. This was a different type, type 1F, so it's different than type 2. But it was the first publication showing, hey, you know what? Some phages are actually carrying proteins that are blocking CRISPR-Cas system. They're called anti-CRISPR proteins, ACR. Right? This is a very hot field right now. So some phage will bypass by just producing protein, and it will go and bind to the CRISPR system, or Cas system. So then there was a bunch of publication related to anti-CRISPR because it became a very interesting, even biotechnological tool. You had enough on and off switch, having a protein where you can tell this in genome editing purposes, hey, stop Cas9, hey, open Cas9, just by using those proteins. So it's become a very interesting field, and it's still a hot field, as you can see for the name of the journals. But I want to highlight two things. One thing. So this was the paper published. I still remember that because we got scoop. This was published during Christmas time. Oh, it was lovely. Put online on December 26th. I can say I remember. But it was published in January 2017. A group by Joe Denomi. Joe working at California, also Canadian. But unfortunately, he's in the US. Things happen sometimes. Anyhow, his group found the first anti-CRISPR protein binding to Cas9. So these were the first one targeting type 2. All right? Published in January 2017. In April, published in April, this group from China, totally unrelated, published a structure on one of these proteins. Isn't it fantastic? Again, a group discovered anti-CRISPR protein, published all the work, all the beautiful microbiology work and biochemistry work. 4 months later, another publication found another group with the structure of that protein. You remember? You know how the publication system works, right? You kind of need to do the experiment. You kind of need to write a paper, submit it, and get it to be and then accepted. It kind of takes some time. This is a lovely field, you know. 4 months. So yeah, it's a very fun field. Anyhow, so in that paper, they found 4 anti-CRISPR protein, all in Listeria prophages, so phage integrated in the genome of bacteria. And so the mode of action is starting to emerge for some of them. This particular one is actually it's binding within the Cas9, and it's preventing recognition of the DNA. So it turned out that not all anti-CRISPR protein are working the same way. So a lot of people are actually studying now to understand how they work at the different levels. Some block the access to guide RNA, some sequester the domain, and so forth. So different proteins, different mode of action, and so forth. And we got scoop, so our ACR became 5 and 6 because the first 4 ones were in the same paper. So we found our first phage, Streptococcus phages, from the industry, from the dairy industry, that were no longer responding to CRISPR-Cas. And they were producing anti-CRISPR protein, different families than those that have been discovered by Joe's group. And Alex Ein and Geneviève Rousseau did the work here. And how we found those? We found those because this is our typical phage assay to get CRISPR BIMs, those colonies that are resistant to phage. But with this one phage, we didn't get many colonies at all. What the heck is going on here? And on top of that, when that phage was supposed to be killed by the spacer in the Cas9 system, the phage was winning. So there was something wrong with this phage, or not wrong, but interesting with this phage, because it was bypassing CRISPR. We could not get new spacer acquisition and so forth. So what we did, we started to clone every single gene of that phage. And then we had to go like 80% because we were not lucky. So what we did is we cloned every single gene, put them in an expression vector, and then we were introducing in a strain carrying a spacer. That phage, which was normally killed by the CRISPR-Cas system, was no longer killed by the CRISPR, so we had to find a gene that was responsible. CAR-28 here became ACR2A6. And that ACR2A6 is actually found in only 5% of the phage genome, but last summer we found this other here, which is found in about 1/3 of the phage genome that we have analyzed carries this CRISPR thing, this anti-CRISPR thing. So what happened here is that we generated in 2005, 6, 7, 8, 9 bacteria that were resistant to phage using CRISPR. Those strains were put into the market, right? And they were sold because they were just natural phage-resistant strains. And now what we're seeing, phage emerging that have ways of bypassing CRISPR. So for us, we're kind of back to the drawing board here. We need to find new natural ways to make the bacteria resistant to phage because the phage, I found a way to bypass. All right. So we saw the structure of one of these in collaboration with Christian Cambio and Adèle Goulet here in Marseille. So that's another reason why I'm here this week. And so seeing our collaborators here. And we're still working on other aspects of the structure of these proteins. And we have shown also with Yannick Douaillon at the Université Laval that the anti-CRISPR also work in human cells. So you can block SP-Cas9 for genome editing purposes. So it works in human cells as well. Cell line. I'm going to skip this. Now what? Well, again, we need to go back to the drawing board. We're looking at different things. One thing you can do is we know that CRISPR-Cas and restriction modification system, when they are together, they work quite well. You can provide an increased phage resistance. So right now, we're looking at different barriers, again, to make the strain resistant to phage. CRISPR and RM are quite useful together. They both cut the DNA, so it's great. But still work in progress. And time is flying, so I'm going to go changing subject here, going to the, so what we're doing now. So we're studying, again, I was trying at the beginning of my talk that we're studying phage biology in my lab. If you take any given phage genome, you look at the sequence of it, do bioinformatic analysis on it. About half of a viral genome, you know what the genes and proteins, what's their function of. But the other half, we have no idea. We have no idea. So these are what we call like a black box or dark matter or whatever. So all these genes are coding for proteins of unknown function. So we're right now starting to producing these proteins, solving their structure, and trying to figure out what their function. So it's a long-term project. We have spent a lot of time looking at the phage proteins that are involved in the structure of these virions here. We've done a lot of work with Christian on these proteins. And now we're working with Adeline to try to figure out what the heck these proteins are doing when the phage infect the cells. And to do that, you need something to modify the phage genome. And it may sound simple, but it's actually quite difficult to do this with a phage. And before CRISPR-Cas9, there was some model of tools, but we're not very efficient. So with CRISPR now, we're using it to modify the genome of a phage. I'm going to skip this right and just show you how it works. So essentially, we will generate in the bacteria, we'll engineer this time a CRISPR array, which will be targeting the gene of interest we want. Let's say there's a gene, we don't know the function. We're going to introduce a spacer that will target that gene inside the phage genome. So that cell will carry this. That cell will also carry a recombination template, another plasmid that will be carrying here homologous region to the upstream and the downstream region of the phage genome. So the phage will infect, it will cut through the CRISPR and Cas9 system, and then the phage again will die, as I showed you before, right? Unless you provide a template for recombination, and what's going to happen is going to rescue this and do by homologous recombination. You will introduce that piece of DNA right here inside the phage genome. And you can knock out the gene, you can add a tag here and so forth. So if the gene is non-essential, you can knock it out very easily. So we have done that with a number of phage gene in the genome of Streptococcus here. As you can see, we have knocked out or deleted some of these genes. We have done this with another system here. Even one phage, we have knocked out 4 different genes, and the phage is still functional, less efficient than the wild type. So you can see the possibilities are just mind-boggling because you can do whatever you want. And that, there's no way you can think about this 5 years ago. So that's why when I'm teaching microbiology, I'm telling our students, you know what? You're so lucky to be in this field right now. Because with all the tools we have, I mean, sky's the limit. Really, it really is. We've got so many tools. You can modify something. You can do omics to death. You can do structural biology. You just need a good idea, hypothesis, and you can add one. It really is. All right, conclusion perspective. Today I might have told you about this much of the field. You would bring somebody else from type 1, different story. You bring someone from type 1E, type 1F, type 3, type 5. They have their own story. Now we know, you can believe that, some CRISPR will pick up the RNA, will pick up spacers from RNA. Not DNA, from RNA. So it's a different CRISPR-Cas system. I mean, it's insane. No, it's fun. I mean, it's not insane, it's fun. But so the system is extremely diversified, versatile, and that's why it's great, because if you're using one system, you know, you can study that system, and the likelihood of getting scoop is still high, but less high, okay? Because there's so many different systems. I hope I've convinced you that inside of bacteria, you can use adaptation. It works by 3 steps: adaptation, expression, interference of DNA. I was briefly mentioning that the industry now today is using CRISPR-ized strains. I keep joking that if you're eating yogurt, you're eating cheese, you're eating CRISPR, you're eating Cas9. That's kind of safe, I would say, because that's what it is. And the strains that are being out there in the market, they have acquired multiple spacers targeting different phage. All right? This is a natural system, so there's no genome engineering here. We're just challenging the phage, the bacteria with the phage, and pick out the right colonies that have acquired. Of course, we're looking at strains that have acquired spacers from genome that are conserved regions in the phage because they have better efficiency and so forth. I mean, there's a lot of tricks when you pick up those colonies and use them. And sometimes people think it's trivial, but you know what? When you're producing something like on the market globally, you need to find a strain that will be efficient in your product, that you're able to produce, that is able to ship all over the place, that you can be stable. It's not that simple. I mean, it's not you go there, pick up a strain, and voila, I'm going to use that. No, there's a lot of work just to characterizing those strains that are going to be used in your food, right? So when you've got a problem like phage, you don't want to throw that strain away because you have invested so much into that strain to characterize it. You want to reuse it and reuse it and reuse it again. But if you do that, you're going to have that phage issue. So it's really important to actually have phage-resistant bacteria. And because phage and viruses are evolving, this is a lifetime job, right? So I'm not going to retire, I'm telling you. I'm going to go. I'm kidding. My wife thinks I will not retire, but anyhow. No. So CRISPR can use to type your strains. Thermophilus is a great example. We're working today with the Canadian government to type Salmonella strains, for example, with the spacers. So let's say, for example, there's an outbreak of Salmonella in Toronto, and there's an outbreak of Salmonella in Quebec. You can look at those strains. Obviously, you can sequence the genome of that strain, but you can just do the PCR on the CRISPR, sequence it, and if you have the same spacer, you know what? Pretty good chance it's the same strain, right? So it's interesting as well. Because of the patent issues, which I will not talk to you about because I don't want to find myself in the bottom of the sea, there's a lot of people that are looking at different Cas proteins, Cas9-like. They have different advantages and so forth. There's a lot of people looking at different Cas proteins that cut DNA or RNA molecule. Cas12a, which used to be CPF1, now it's called Cas12a, and Cas13, which is targeting RNA, not DNA, like I was showing you. Targeting RNA, all these different proteins that are being studied, and the application of CRISPR-Cas9 are unbelievable. You can do base editing now, you can do DNA imaging, because you can go and target whatever you want. You can put fluorescence in there, it's going to go and target that gene. You can do gene knockout, because you can, Cas9 has 2 domains that cuts the DNA. You can knock out those domains, so it won't cut anymore, but it will hide the RNA, so it will bind to DNA and stay there. Yeah. So there's no more transcription. So you can, regulate the expression. I mean, sky's the limit with these things. It's unbelievable. This is just a tiny, tiny thing related to CRISPR. And that's why I was telling you at the beginning of my talk, this is a career opportunity in the sense that you should know about this system. You should inquire how it works in your field, because it's becoming a tool very useful in your research. And I'm going to finish on this. For us, it's pretty amazing because we really started with an applied problem. The dairy industry was having a phage issue. It still has a phage issue. And we wanted to have bacteria that are resistant to phage. and now some bright people have used this to make maybe this fantastic tool now in basic research. And on that, I want to thank people that were involved in this research. Josiane, Manuela, Marie-Ève, Alfonso, Hélène, Bruno, Alex, Marie-Laurence, people in my group that work on CRISPR-Cas, and there are a few others now. We have been working in collaboration for almost 20 years with DuPont. I do not have any stocks in the company, but they are really nice collaborators. They have been funding my lab, and I'm very thankful to them. And they have become friends now, essentially, because we have been working together. Sometimes you hear these stories about industry, university collaboration, that they don't work, and blah, 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 and you have these horror stories. But this has been a great, great, great collaboration with them, and I want to acknowledge that. And on that, I want to present you my group here, and our website is pretty much up to date. We're a phage lab, doing CRISPR work, but doing a bunch of other phage stuff. So if you want to know what else we're doing, you can go to our website. And of course, I want to thank all our funders, because without them, there's no research. So I want to thank them as well. I want to maybe highlight Denise Tremblay here, which is a lab manager in my group and also in charge of phage collection. She's been with my group for 20 years now. And also Geneviève Rousseau, another researcher that has been in my group for 15 years. And on that, I want to thank you all. And please, if you have any question, do not hesitate. Et merci beaucoup.
I think that Emmanuel, to start with, was in the lab of Patrice Corona, is that right?
Oui, exact.
And you know, Patrice got a very nice question that we are working hard with Jean-Marc on, is that he say, well, I'm not surprised that bacteria become resistant to antibiotics. I'm interested in the fact that some bacteria never keep the resistance. And since this time, we think about this, it was in my scientific committee at the beginning, that probably it's because the immune system of Streptococcus are not caring for a very long time for DNA. Including probably, I suspect that if scarlet fever, if it disappeared, is probably because of a CRISPR or CRISPR-like system, because it's a phage.
But I think it's also, it depends on the species, because Streptococcus pneumoniae does not have CRISPR, but it's naturally competent. So in the case of pneumonia, it needs to acquire a piece of DNA from the environment to become more efficient into the ecosystem. But then CRISPR would actually be detrimental to that. So even if we introduce CRISPR, and some work has been done at Rockefeller University, if you introduce CRISPR, pneumococci just get it out.
Because because it's foreign.
Yeah, that's right. Well, foreign. Yeah.
Well, I'm not sure. I think even for pneumococcus, it's very bizarre that pneumococcus have not really, of course, people like to say that it is resistant. In fact, it's not when it to beta-lactam. So I wonder why this bug has been exposed for so long. And it is partly, not uniquely, but partly a human commensal. So it's been in contact with beta-lactam for a very long time, and it did not acquire resistance.
I think that's a great question regarding antibiotics, but I think regarding phage, pneumococci is interesting because of the capsule, and that in pneumococci is very, very efficient against phage. It's, even though pneumococci is loaded with prophages, try to find phage that will infect a pneumococci actually pretty hard to do. And again, the capsule is diversified. There's so many serotypes and so forth. In that particular bug, it looks like capsule is pretty important and probably provides some selective pressure back. It's one of those things, but yeah.
Oui. Je vais poser en français. Vous avez montré qu'il y avait CRISPR, il y avait anti-CRISPR.
Oui.
Après, il y avait. Et je me demande si les bactéries vont pas inventer des anti-CRISPR.
Il y a
des anti-CRISPR.
Ben, c'est-à-dire qu'on pense qu'il y en a.
On pense. Tout je sais.
Oui, il y a un papier qui sortit sur les anti-CRISPR.
Non, encore.
Ouais. Je l'ai déjà lu.
Non, mais voilà,
mais il y a d'autres mécanismes. Oui, mais il y a d'autres mécanismes de résistance aussi. Hein. Donc, CRISPR, c'est il est efficace, pas tout le temps. Puis, en fait, on s'aperçoit maintenant qu'il est aussi très silencieux dans d'autres espèces bactériennes. Streptococcus, c'est peut-être un des plus beaux systèmes, parce que les récepteurs, les récepteurs de phage qu'on ne connaît pas en passant, pour l'instant, c'est le CRISPR qui est vraiment dominant. Mais dans d'autres espèces bactériennes, dans E. coli, vous savez...
Enfin, moi, ce qui m'intéresse, c'est le côté universel. Les CRISPR apparaissent de façon indépendante, parce qu'il y a des familles complètement différentes. D'accord. Les anti-CRISPR eux-mêmes, c'est des trucs complètement différents.
Exact.
Et donc, c'est des systèmes très, très universels, très dynamiques. Et comme dit Didier, ça ne m'étonnerait pas qu'il y ait...
Oui, je suis d'accord avec vous.
C'est fantastique, en tout cas. Bravo.
Est-ce que vous avez déjà vu des systèmes de transposons dans des plasmides réplicatifs conjugatifs qui concerneraient quelque chose qui est un gène, flanqué par des transposases, donc un transposon qui contiendrait les anti-CRISPR?
On ne peut pas regarder ça
tout à l'heure.
Il a ça.
Il a ça.
C'est sûr, mais c'est un mécanisme qu'on pouvait imaginer. Vous pouvez imaginer que tu as une transposase qui vient avec un anti-CRISPR pour neutraliser CRISPR. On peut imaginer.
Oui. Dans quelle mesure la mémoire immunitaire persiste sans exposition répétée? Est-ce qu'il y a une accumulation permanente ou il y a des systèmes de remise à zéro?
Écoutez, dans nos mains à nous, chez Streptococcus, c'est très stable. Donc, on propage la bactérie, puis on n'a pas de perte. Toutefois, il nous est arrivé à l'occasion, lorsqu'on générait une bactérie qui était résistante au phage, donc elle avait acquis une belle espaceur, on avait effectivement des délétions, mais dans la partie centrale du CRISPR array. Donc, dans le milieu, on perdait 4, 5, 6 espaceurs. C'est comme si la bactérie se disait, moi, je n'ai plus besoin dans mon environnement, là, puis j'enlève ça. Et puis, mais jusqu'à date, on a manqué de phage. À un moment donné, de ce qu'on arrive, on n'a plus de phage. Non, on n'a pas vu de limite. On a fait des études, je pense qu'il y avait un fitness cost, un coût. Puis l'ajout de 66 paires de bases, pas vraiment, même s'il y en a plusieurs. Par contre, il y a un coût un peu plus important quand la bactérie se défend. Donc, quand le phage infecte, évidemment, puis là, il faut se défendre, là, il y a un petit coût là, énergétique. Mais sinon, non, il n'y a pas de... On n'a pas vu ça. Donc, des délétions uniquement quand il y a une infection. Puis la délétion se faisait tout le temps concomitamment avec l'ajout d'un espaceur, aussi en 5'. Voilà, c'est tout ce qu'on a vu. Mais nous, c'était dans nos mains. Encore sur le Streptococcus, là, c'est stable.
S'il n'y a pas de système d'élimination, en théorie, il y aura des accumulations lorsqu'on a l'exposition.
Mais vous savez, je pense qu'on n'a pas déterminé. Souvent, chez les streptos, la souche native va avoir une vingtaine, une trentaine d'espaceurs. Je pense qu'on a déterminé jusqu'au 14e que c'était efficace. Après, je ne sais pas si l'avant-dernier est encore bon. Je ne sais pas. Ça, on n'a pas fait.
Et sur le même sujet, est-ce que la séquence des spacers permet de faire de la phylogénie des phages en termes d'évolution?
Je n'ai pas vu. Mais ce qu'on a vu, par exemple, c'est dans les analyses de métagénomique. Donc, les analyses de métagénomique, si on trouve un CRISPR array, on peut prendre les espaceurs, sortir ces espaceurs-là, et aller associer ces espaceurs-là à un contig. Et là, dire, ben ça, c'est un contig probablement viral. Peut-être plasmidique, là, mais c'est probablement viral. Ça, on a vu ça. Mais ce que vous dites, à part ça, je n'ai pas vu. Donc, on sert des espaceurs pour même dire, c'est peut-être, le phage est peut-être même trouver son hôte. Donc, on a un contig viral dans un métagénome, on a un espaceur, exemple de colis, ben, ce virus-là, c'est peut-être un phage de colis, ou un plasmide de colis. Ouais. Donc ça peut se faire.
On a de théories, on a beaucoup, on en change volontiers aussi. Mais moi, j'ai une théorie sur l'apparition des clones, des clones épidémiques. Et je pense que très souvent, les clones épidémiques arrivent par le knockout de CRISPR. C'est-à-dire que quand on knockout de CRISPR, c'est ce qu'on voit dans pratiquement tous les clones épidémiques. Les clones épidémiques ont une susceptibilité aux gènes de résistance qui est beaucoup plus importante.
Oui.
Et qui n'est pas seulement expliquée par le niveau de multiplication. Et c'est vrai pour tous les clones épidémiques.
Ah ouais.
Donc, j'ai vraiment l'impression que les clones épidémiques ont à la fois une perte de gènes très importante. Ils sont plus petits. Les clones épidémiques, l'histoire des gènes de virulence, c'est une connerie, c'est pas vrai. les jaunes épidémiques sont plus petits que les espèces sources.
Ouais.
Un tiers de gène en moyenne, hein, pour les bad bugs, que moi j'appelle les 12 bactéries les plus terribles de l'histoire de l'humanité, sont toutes 30% plus petites que leurs voisins. Mais dedans, ce qu'on n'a pas regardé à l'époque, c'est en même temps qu'elles commencent à perdre de leurs chromosomes, si elles ne perdent pas les systèmes d'immunité contre les gènes aliens, et si ce n'est pas à la fois la perte des gènes et la capacité d'acquérir des gènes aliens qui fait qu'elles sont à la fois très rapides et à la fois résistantes. Et qu'on ait tous les trucs que Parkill fait, où il fait 1000 bestioles, les 1000 clones, on voit que dans les clones, c'est les clones, comme que ce soit Beijing, ou que ce soit typhoïde, ou ça, c'est les clones qui sont épidémiques, qui sont aussi susceptibles d'acquérir des résistances par transfert latéral de gènes. Donc j'ai vraiment l'impression que le système d'immunité contre les gènes aliens a disparu en même temps qu'une partie.
Très possible. Il y a, pour 2 points sur ce que vous venez de dire. La première, c'est... je me souviens d'une étude qui était faite au Texas, ou même, non, pardon, c'est à Harvard, où ils avaient démontré qu'effectivement, chez les entérocoques, il y avait vraiment une corrélation chez les entérocoques. Tu avais un CRISPR, tu n'avais pas d'antibiotiques.
Oui, c'est un homicide.
Exactement. Et le deuxième point que je veux dire, c'est que, j'en ai pas parlé dans ma présentation, mais ça arrive à l'occasion que la bactérie va acquérir un espaceur contre elle-même, qu'elle fasse de l'auto-immunité. Parce qu'elle peut acquérir un espaceur contre le phage, contre un plasmide, mais contre son propre génome. Et dans ce moment-là, qu'est-ce qui arrive? La bactérie meurt, ou elle va muter sa cible, ou elle va carrément l'enlever, ou bien c'est le CRISPR qui va partir, parce que sinon c'est la mort de la bactérie. Donc ça arrive, mais c'est des phénomènes qui sont très peu fréquents. Donc l'adaptation, je ne sais pas si vous l'avez noté, là, mais l'adaptation c'est un phénomène très rare. Hein. La plupart des cellules qui sont infectées par un phage, ils meurent. Seulement quelques-unes vont survivre, celles qui sont infectées par des phages virulents, mais le processus d'acquisition d'ADN, l'adaptation, il est très peu efficace. C'est normal, ça. Sinon, on saute trop dessus elle. Il y a un phénomène accéléré d'auto-immunité, en fait. Là. Et ça, donc, c'est un fine-tuning, finalement. Moi, ça me plaît.
J'ai encore une question.
Oui.
Je reviens, les bactéries du vinaigre, elles sont résistantes au phage, je pense, sinon...
Les bactéries du vinaigre, là, ça, c'est une grosse job.
Donc, je veux dire, est-ce que toutes ces bactéries de l'agroalimentaire, elles n'ont pas été, comment dire, sélectionnées parce qu'elles étaient résistantes au phage?
Non, on a la bactérie alimentaire ou laitière, par exemple, la plus importante, c'est un lactococcus. Il n'y a pas de CRISPR. Parce que le lactococcus, qui se développe dans le lait, a besoin beaucoup de gènes auxiliaires pour se développer bien dans le lait. On les a sélectionnés avec le temps pour avoir des plasmides, parce que plusieurs copies du même gène, donc sont très performantes dans le lait. Donc on a sélectionné des bactéries, on les a domestiquées, puis en faisant ça, on a sélectionné des bactéries qui avaient plein de plasmides, parce qu'il y a des gènes qui sont foutument importants, et le nombre de copies est important. Et donc eux, ils ne veulent pas de CRISPR. En fait, il n'y a pas de CRISPR dans les lactocoques.
Elles sont résistantes au phage?
Ben, par d'autres moyens.
Comment?
Par d'autres moyens.
Voilà.
Ouais.
Donc ce que je veux dire, c'est
que s'échangent des plasmides.
Toutes les bactéries de l'agroalimentaire, elles sont résistantes au phage d'une façon ou d'une autre. On a sélectionné ça.
Bon, on espère. Pas tous, là. Mais ça, vous savez, la notion de résistance au phage, là, ça n'existe pas. Là. Il y a toujours un phage qui va arriver. Ça, l'industrie.
Jusqu'à maintenant.
Oui, mais il y en a toujours. Ça fait...
Je suis d'accord, c'est pour ça que j'aime bien, je me méfie des sujets qui réémergent trop rapidement. Et donc, c'est vrai qu'il y a beaucoup de choses sur la Patrick Forter. Je ne crois pas beaucoup à la phagothérapie parce que ça a déjà beaucoup échoué, parce que la vitesse d'acquisition de la résistance, c'est beaucoup plus rapide que la résistance aux antibiotiques. La résistance aux antibiotiques, c'est compliqué. La résistance aux phages, c'est intrinsèque aux bactéries. Donc, découvrir des bactéries résistantes aux phages, c'est extrêmement rapide. Donc, c'est pour ça que la phagothérapie, je suis extrêmement réservé sur la phagothérapie.
Je contentais.
Oh, oui. Là, je vais vous donner un exemple des fois, parce qu'on travaille beaucoup avec l'industrie laitière, puis des fois, ils me disent, bon, ben, j'ai trouvé un phage dans mon usine, puis je veux utiliser cette bactérie-là, donc c'est quoi mes options? Ben, les options, c'est faire un peu ce qu'on a fait, ou changer carrément de bactérie. Vous savez, la configuration de la salle fait que si vous parlez en haut, hein, on vous entend en bas. J'enseigne, moi, je suis au courant de ces phénomènes-là. Bon. Oui, c'est ça. Donc, il y a une façon un peu naïve de penser, c'est que je vais retirer ma souche d'intérêt et je vais la réintroduire plus tard. Eh bien non. On a fait un essai à un moment donné avec une entreprise où il y avait un phage important. Ils ont retiré la source, la bactérie. 14 mois plus tard, ils remettent la souche, le même phage. On a pris le phage, on l'a séquencé, on l'a comparé. 14 mois plus tard, c'est le même phage. C'est donc, de se débarrasser d'un phage dans une usine, dans un environnement, bonne chance. Et ça évolue en plus. Non, mais c'est fabuleux! On a du travail! Non, j'exagère, là, mais ça peut être un problème. Mais on a quand même des biocides. Dans le domaine alimentaire, on a quand même des biocides et tout ça, on peut se débarrasser de d'autres façons. Là, c'est fabuleux parce que l'industrie a des procédés maintenant qui est super intéressant où on essaie de récupérer des protéines de lactosérum, en tout cas des sous-produits qu'on veut valoriser, puis on fait des traitements thermiques pour réintroduire ça dans le lait, c'est super beau. Puis là, en faisant ça, ben, on est en train d'introduire des phages qui sont thermorésistants. C'est fabuleux. Les virus, c'est fantastique. Bon, voilà.
Non, mais la vie, c'est la guerre.
Ah oui. Oui, c'est ça, la guerre.
C'est la guerre.
Est-ce qu'il y a d'autres questions? Oui, à l'arrière.
Vous avez parlé principalement des phages virulents.
Oui.
Les phages tempérés, ça se passe un peu comment en fait? Est-ce qu'ils ont plus d'anti-CRISPR? Est-ce qu'ils arrivent mieux à contrôler, par exemple? C'est pas bon ça.
Oui, c'est une bonne question. En fait, les premiers anti-CRISPR ont été trouvés dans les prophètes. Donc, ça c'est clair. C'est sûr que quand il y a une induction, ça peut bloquer, effectivement. C'est des joueurs importants, particulièrement les bactéries pathogènes, parce qu'il y a beaucoup de prophètes qui peuvent pour les bactéries vivants. Mais oui, ils sont susceptibles au CRISPR aussi, quand ils sont induits. D'accord.
The protospacer adjusting motif that you have shown.
Yeah.
So is it just the one, or there are multiple type of bands motif? And I would like to know whether they are either strain specific or specific to a Cas system.
It's a great question. Yeah, so I was trying to say that each PAM is Cas9 specific. So depending on the Cas9 you're dealing with, it's going to have its own PAM. So in the Cas9 protein, it's a big protein. The C-terminal part of the protein has what we call a PAM interacting domain. So that sequence there is very important to recognize. So there's specific amino acids that will recognize those nucleotides. So it's really specific. So different Cas, different PAM. People have actually used even Cas9 pyogenes and do a random mutation, and you can generate even different PAM using the same protein just by evolutionary modification. So yeah, but it's really one part of the protein that is responsible for that.
So its size is restricted to 4 to 6 base pair, or it can be a bigger one?
Yeah, it's usually 3, 4, 5, 6. I think there's a few bigger ones, but not many.
So is there a database where we can find this shortlisted?
No, they're called reviews. But it's a good question because the PAM is really important. Because if you're like NGG, I mean you need to have a GG. Human genome, not that many, right? So sometimes it's good to have an AT rich PAM and things like that. So yeah, there are a lot of people looking into that because there's obviously patents and stuff. But the patent is not a big deal for us because you can use whatever, for research you can do whatever you want. So, I mean, you should not even think about that. If you want to have, you have an idea, you want to use the tool, just go ahead. Now, if you want to develop a product or you want to have a therapy at the end of the day, that's a different story. But for your research purposes, it's not a problem. It's fantastic though. So that's why, we got all these, we did not talk about that because I don't want to. But all these engineered babies or whatever, I mean, that's a totally different story. But for us as a scientist, the tool itself, it's fantastic. Think about it. You got a genome, you want to modify something. Instead of thinking, okay, maybe I'm going to do this mutant, it's going to take me, I don't know, 6 months, 9 months, depending on what system you're working. Now you can do it. It's going to work like that. Now, so you can ask yourself different questions because you know you're going to get your cell, you're going to get your mutant, you're going to get it. If you do it right, you're going to get it. You can start doing, you can do multiplexing, you can knock out a bunch of things together. I mean, sky's the limit. It really is. It really is. Sometimes people are like, oh, whatever. I don't know. It's not whatever. It's happening. But I'm not a eukaryotic person.
Okay, thank you.